Good morning, students. In my previous video, I have already explained these terminologies. First one is this one: alleles, factors, characters, traits, etc. So now moving on to continue this terminologies. Phenotype. Phenotype, it is the observable characteristic of an individual, as for example tall or dwarf. That means those characters that can be seen externally of an individual is called phenotype. What is genotype? It is a gene complement of an individual with regard to one or more characters, such as TT, TT. So these TT, TT, TT are the tallness or height of the, it refers to the height character of the individual. So this is internally related, and so it is called genotype. Now come to the homozygous. Homozygous, it is the individual having identical alleles of genes such as TT, the individuals which contain the similar alleles, that is capital T, small t, small t. So those individuals are called homozygous. Heterozygous, an individual having both the contrasting alleles of character is called hybrid or heterozygous individual, such as T and T. Suppose if we cross between the two individuals of character height, then if one is tall and another one is dwarf, then their gametes will contain T and T, and its gamete will contain small t and small t. When these two characters again these two gametes again fuse together to produce this type of character or individual containing capital T, small t, then we can say it as an hybrid. This is called hybrid with genotype capital T and small t. Okay. Now you can see dominant factor. So what is the definition? It is a factor or allele which can express its effect whether present in homozygous or heterozygous state. So we can say in this figure, suppose this is tall and this is dwarf, and tallness is the dominant character and it appears in the hybrid. So hybrid, it is a tall, the hybrid that produce is called tall. So tall is the dominant character, and small t, that is dwarfness, the alleles of the dwarfness does not appear in the hybrid, so it is the recessive character. Then F1 generation, the full form is first filial generation or son and daughter generation. How can we know this one? It is the generation of hybrids produced from cross between between genetically different individuals called parents. Suppose we are in the first filial generation of our parents, and if our daughter or son may be the second filial generation of our parents. Okay, then second one is F2 generation, that is F2 or second filial generation is the generation of individuals which develops as a result of inbreeding amongst individuals of F1 generation. A monohybrid ratio. It is the ratio obtained in F2 generation when a monohybrid cross is made and the offspring of F1 generation are self-bred. Monohybrid ratio is 3:1 phenotypic ratio, and 1:2:1 is genotypic ratio. So what is monohybrid cross? Monohybrid means the cross between the individual with a single character. So this type of cross is called monohybrid cross, and ratio obtained during this cross is known as monohybrid ratio. And likewise dihybrid ratio, it is the ratio which is obtained in F2 generation when a dihybrid cross is made and offspring of F1 generations are self-bred. Dihybrid ratio in 9:3:3:1. This is phenotypic ratio, and 1:2:2:2:4:2:2:1:1 is the genotypic ratio. That means what is the dihybrid cross? The cross in which two characters of an individual is involved, such as suppose if we take the pea plant, then suppose it's round seed and green seed. These are both two characters, so round and green crossed with the yellow and wrinkled. Okay, so these two characters are involved, so this is called dihybrid cross, and the ratio obtained by such cross is called dihybrid ratio. Okay, now Mendel was educated in a monastery and went to the study science and mathematics at the University of Vienna. Failure in the examinations for teaching certificate did not suppress his zeal for scientific quest. Zeal means jazba. He went back to his monastery and started growing peas. Peas means matar. Many others had studied the inheritance of traits in peas and other organisms earlier, but Mendel blended his knowledge of science and mathematics and was the first one to keep count of individuals exhibiting a particular trait in each generation. This helped him to arrive at the laws of inheritance. Further on, these laws of inheritance is renowned by us as Mendel's laws of inheritance. And who was Mendel? Mendel was known as the father of genetics. So what is that laws? Mendel used a number of contrasting visible character of garden peas. These characters, you must learn these characters, that is round wrinkled seeds, tall short plants. These are the contrasting character, and taking these pairs, Mendel started his experiments. Tall short plants, white violet flowers, and so on. He took pea plants with different characteristics. A tall plant, a short plant, produced progeny from them and calculated the percentage of tall or short progeny. Why did Mendel choose the pea plant? Because it is easily available everywhere. That's why Mendel chose this one, and it is a bisexual plant. That means male and female both parts are present in single flower. That's why it is easier to obtain all the things within a single flower. So he chose it, and so many varieties, that is so many pairs where Mendel can see in a single plant. That's why pea plant is chosen by Mendel. And this one in the first place, so this is how can he evaluate the inheritance of traits over two generations. So in the first place, there were no halfway characteristic in the first generation of F1 progeny. No medium height plants or plants were tall. This meant that only one of the parental trait was seen, not some mixture of two. So the next was seen. So the next question was, were the tall plants in the F1 generation exactly the same as the tall plants of the parent generation? Mendelian experiment test this by getting both the parental plants and these F1 plants to reproduce by self-pollination. The progeny of parental plants, of course, of all tall. However, the second generation or F2 progeny of the F1 tall plants are not all tall. Instead, 1/4 of them are short. This indicates that both the tallness and shortness traits were inherited in the F1 plants, but only the tallness traits was expressed. Thus, two copies of the traits are inherited in sexually reproducing organism. These two may be identical or may be different. Depending on the percentage, a pattern of inheritance can be worked out with this assumption as shown in figure. So, you can see here tall and short, first pair. He crossed between the two, and all the in F1 generation, all the tall plants are all tall, you can see, but the traits appear is capital T and small t. That means the alleles from both the plants are inherit in the F1 generation, and again when these both the plants are self-pollinated, that is TT and TT, that is both this type of plant, two plants are crossed between or self-pollinated, then what happened? That means three, these are the progenies of the F2 generation are in the ratio 1 to 3. 3 are the tall plant and 1 is the small. So 3 is to 1. So we can make a cross through only taking their genotypic character. Okay. So you can see here that how Mendel's law of inheritance can be proposed. So it is a monohybrid cross. Why? Because a single character will be taken. Okay. So tall and short plants. So when these two plants are crossed together, then the hybrid will be T and T. That is, all the plants will be tall in F1 generation. Okay. And also phenotypic appearance is tall, but genotype is different, that is capital T and small t. That means the alleles from both the what is allele? This T and T are alleles. So alleles from both the parents are inherit in the F1 generation. And when these F1 generation are self-pollinated or reproduces among themselves, the gametes are here TT and TT. Both have similar gametes. But in the F2 generation, this is called F2 generation, or that is second filial generation. What we gain when T, again capital T, small t, and this when it fertilizes this, then small t, capital T, and this fertilizes this, then small t, small t. So the progeny means the offspring, the child. So the progenies of the F2 generation are 3 among them are tall and 1 among them is short height. That means this is phenotypic, and phenotypic ratio will be 3:1. But the genotypic ratio is 1:2:1 in F2 generation. How it appears? That you can see here T and T. Okay, this is one only. Capital T, small t contains 2, and small t again 1. So it is 1:2:1. So this is called genotypic ratio. And now you can understand this, that it is in F1 generation tallness is appeared, so it is dominant character. Okay, everywhere tallness is appeared, it is dominant character, and this is the dwarf allele, so it does not appear, so it is the recessive character. Okay. In this explanation, both capital TT and capital T, small t are tall plants, while only it is a short plant. In other words, a single copy of T is enough to make the plant tall, while both copies have to be small t for the plant to be short. So traits like T are called dominant traits, while those that behave like small t are called recessive trait. Work out which trait would be considered dominant and which one recessive in figure 9.4. You can see here this figure which one is dominant. You can see here purple color is dominant and this green color is recessive. Okay. Now what happens when pea plants showing two different characteristics rather than just one are bred with each other? What do the progeny of a tall plant with round seeds and short plant with a wrinkled seed look like? They are all tall and have round seeds. Tallness and round seeds are the dominant traits, but what happens when these F1 progenies are used to generate F2 progeny by self-pollinated? A Mendelian experiment will find that some F2 progenies are tall plants with round seeds and some were short plants with wrinkled seeds. However, there would also be some F2 progenies that showed new recombinations. Some of them would be tall but have wrinkled seed, while others would be short but have round seeds. You can see as to how new combinations of traits are formed in F2 offsprings when factors controlling for these shapes and seed color recombine to form zygote, leading to form F2 offspring. Thus the tall short trait and the round seed wrinkled seed traits are independently inherited. Okay, so in this video till this I will explain next topic in my next video. So watch this video. I hope you can understand quite well all the terminologies and of course the monohybrid cross of Mendel's. Okay, thank you.